The National Center for Science Education also engages with communities, eager to interact with scientists, and they block attacks on science education. So it's my pleasure tonight to welcome Dr. Reid to tell us about the 1918 flu. Dr. Reid.
All right, thank you. Let me just correct you that I am not actually a doctor.
Are you able to screen
occasionally
share?
play one on TV. So let me share my screen. So that's going to stop the other screen sharing. Is that okay?
Yeah. Yes.
Okay. All right. Give me a second here.
It should, yes.
And let's make sure that I do this right, because we worked on this yesterday.
While she's doing that, this is Patricia. Let me just say why you're not seeing me is because I have a nosebleed that just won't stop. So I'm afraid that all of the confusion is because of that. So my apologies.
Oh, I'm so sorry.
Always something, right?
Patricia promises to be back Saturday night as host of this series.
It ought to be done by then.
We hope.
We'll be talking about the history of nosebleeds.
Yeah.
We'll
It'll be a record nosebleed by then, probably. All right, here we go.
Okay, sorry for a slight ride on the struggle bus. Thank you so much for including me in your speaker series. I really appreciate it, and I'm going to have to come back for the puppy talk because I'm a huge dog fan. Working on this talk for today has really taken me back in time and made me realize how different the saliency of studying the 1918 flu was back in 1995 versus how I feel about it today. Obviously, being in the middle of a pandemic to rival the 1918 flu, which I am feeling, looking back at what we were thinking in 1995, between 1995 and 2004, what we were concerned about then, all of which was legitimate, but what we were not maybe concerned about because this is maybe unimaginable. So anyway, we're going to look back at 1918 from the 1995 to the best I can reconstruct how the world was 25 years ago. And then we'll take a little look at it from today. So as you know, science is a noun in the dictionary, but in reality, science is a verb. Science is something that we do. And it's not something static. It's not really a matter of what we know, even though often we think of it, we're taught it in school as if it's something that's contained between two textbook covers. That's not really what the practice of science is. The practice of science is what we don't know. So a key part of science then is engaging deeply with what we do know to identify what we don't know. And that's what we really want to explore as scientists. And what that means on a very practical level is engaging with evidence. So just a brief side comment on the National Center for Science Education. We believe that everyone, all students, deserve to engage with evidence responsibly and directly, especially on topics where the level of misconceptions and the level of misinformation is enormous. And so students are very likely to come into the classroom with misconceptions. And we work very hard to help teachers help those students engage with evidence so that they can correct those misconceptions. So what the current situation is making me think about, though, is how do we decide what of all of the things we know is the most useful? We knew in 1995, we knew a lot about the 1918 flu, although it turned out we had to dig pretty deep to find out how much was actually known about it. But then you have to decide, what should you pay attention to? What is the most important thing to figure out? And that's where I think it's interesting to look at what we thought we needed to learn from the 1918 flu in 95 versus what we now know we still need to learn from it. So of course, you all know that science doesn't start from scratch. You always are jumping into the middle of a long running story. Whenever you do science, there's always a lot of science that's been done before. So I jumped into the influenza story in 1995. And I just thought you might not remember 1995 too distinctly, especially if you listen to a lot of that guy in the middle. 1995 was the year that he passed away. It was the year of the O.J. Simpson trial. It was the year that deaths from AIDS peaked. It was in 1995, Amazon.com was still advertising itself as the world's largest bookstore. They mostly sold books then. And if you were thinking that was so long ago that they only had flip phones, no, we didn't have flip phones yet. And the first flip phone was in 1996. So that's just a little context of what the world was like in 1995. Oh, and AOL.com was the way you got to the interwebs. In science, what was going on in 1995 was, at least in one tiny little corner of science where I was, we were building on a paper that came out in 1985 by Kary Mullis, for which he eventually won the Nobel Prize, describing the technique of polymerase chain reaction. And I remember distinctly being at a journal club in 1986 when this paper was presented to a bunch of molecular biologists, and everybody kind of slapped their forehead and thought, oh, that is so obvious. I should have thought of that myself, because the technique was building on a very basic biological process. And it was pretty obvious once Kary had the original insight. But the other thing I remember about that journal club is all of these molecular biologists sitting around the table going, oh, but it's going to be so tedious because you got to move the tubes from one water bath to another and then open the tubes and put polymerase in, and it's just going to be a mess. I mean, like, all our graduate students are going to be doing is moving tubes back and forth between water baths. Well, it didn't take long for some engineers to get involved and build thermal cycler machines that could do that temperature cycling for you. And then it wasn't very long before a microbiologist identified a polymerase that could survive being heated to near boiling. And then the technique became really easy to do and quick. And it is still enormously important technique. In fact, many of the coronavirus detection assays depend on PCR. So in 1994, the National Center for Bioinformation opened its first website. That's an institute at the National Institutes of Health. And that's where GenBank could be searched remotely. GenBank was the total collection of all genetic sequences that had been sequenced at that time. If you didn't have access to the web, which many of us did not, you could also get GenBank on CD-ROMs that they would send you in the mail every quarter. The entire world collection of DNA sequences in 1995 fit on 5 CDs. In 1997, PubMed launched, where you could search for journal articles online. And then in 1998, Los Alamos opened an influenza sequence database, which was enormously useful because you didn't have to search through all the mouse and yeast and human sequences. You could just gain access to the influenza sequences. So just in that 10 to 12 years, you can see that the stage was set for actually being able to do a project like sequencing the 1918 influenza virus. So what else was going on in 1995? Well, Bill Clinton was president, Al Gore was his vice president, and they had a long-running project that they called Reinventing Government, where they were looking through every department, every agency, and streamlining and getting rid of functions that they didn't think the government needed to do anymore. And so that was an ongoing project, and I was at the time working at the Armed Forces Institute of Pathology, which was part of the Department of Defense. And so that was part of this review of reinventing government. The other thing I was doing was raising these 3 kids. That was their first day of school in 1995. my middle son there was extremely disappointed that he was not allowed to take his plastic sword to school. So the Armed Forces Institute of Pathology is now closed, but its functions have been moved over to the Naval Hospital. But when I worked there, it was on the Walter Reed campus in Northwest Washington, DC. This is the beautiful building it was contained in. That building was built to be an atomic bomb shelter for the White House. So you can imagine the ambiance there is really, really special. But the part of the AFIP that I want to talk about today, and there are a lot of great things about the AFIP, it had a storied history, but one of its, what it considered its crown jewel was its biorepository, which you can see there, those racks that look sort of like the end of Raiders of the Lost Ark. Each of those boxes contains hundreds of formalin-fixed paraffin-embedded tissue samples from every disease you can imagine. That these samples were sent to the AFIP, to its staff of highly specialized pathologists when the diagnosis was unclear. And you got your diagnosis back and the AFIP kept the samples, and there were in 1995 about 2 million samples in that biorepository. However, that Reinventing Government project sort of cast its eye onto that biorepository, asking whether this was really a function that the federal government should be doing. It's expensive to maintain all those samples. Should they keep it? So my colleague, Jeff Taubenberger, and the museum staff at the AFIP had a conversation about what project could be done with those samples from the repository that would prove the value of keeping that biorepository. And they landed on the idea of the 1918 flu. And I remember Jeff coming back to the office and saying, coming back to the lab and saying, we're thinking maybe the 1918 influenza. I don't really know that much about it. I do remember it being mentioned when I was in medical school, and I had never heard of it. And that sounds kind of crazy now when everybody's talking about it all the time. But when we went to search for information about it, this is the only thing that we could find. One book, America's Forgotten Pandemic. And it was written by a historian, Alfred Crosby, who is better known for his work on ecological imperialism. He did a lot of work on the result of colonization and the transfer of germs and crops and animals across continents because of increasing world travel. But he also wrote this book, America's Forgotten Pandemic, which is, I still think, the very best book on the flu.
Click on the little arrow. I think you can.
So we're.
Sorry, does somebody have a question?
No, they accidentally unmuted themselves.
Oh, okay. So should I go on?
Please, it's fascinating.
Okay, terrific. We invited Dr. Crosby up to the AFIP to give a talk in about 2000, I think, 2001. I don't remember the exact date. And we had a million questions for him because we had read this book, you know, like the Bible. Every page was annotated, and he had written it. I can't remember when it came out, 1970 something.
See that little
maybe?
arrow there? That little.
And he couldn't remember anything. He'd written this book so long ago, he really didn't remember that much about the pandemic, which was very disappointing to us. But it's a fabulous book. I just took a couple of photographs of the table of contents and the lists of graphs and tables. He systematically went through everything that was known about the flu and turned it into this wonderful book. The narrative is gripping and his writing is beautiful. And we just went back to it over and over and over again as we came up with questions about what happened in 1918 that would help us figure out what we should be looking for as we start looking for the virus in these samples. So we had a bunch of practical questions. The influenza virus, like coronavirus, is a single-strand RNA virus. and RNA is typically much more fragile than DNA. We had a lot of experience isolating DNA from formalin-fixed paraffin-embedded tissue, but we had not worked with RNA before. So we didn't know if it would even be preserved. If it was preserved, we didn't know how deteriorated it would be, whether there would be any pieces big enough even to do PCR on. We didn't know whether any of the cases from the archive would still have virus, would have still had virus present when the patient died. So there were 75 or so cases of the 1918 flu in the AFIP archives. And Jeff Taubenberger, again, who is a pathologist, went through the cases to try to identify people who had died within a few days of becoming symptomatic. The vast majority of people in 1918 who died of bacterial pneumonia. Only about 2 or 3% died of an acute viral pneumonia. And those would, of course, be the only cases we would have a prayer of finding the viral genetic material in. And we didn't know whether any of the cases would still have virus present. We needed to figure out what subtype the virus was likely to be in order to design our PCR probes. And we needed to figure out which parts of the virus would be likely to be most highly conserved, because that would be our best chance of, sorry, we've gone to a different view. Do you guys still see my screen?
I think you're gonna have to reshare it.
Okay. There. Now can you see it?
Perfect.
Okay, good. The influenza virus has 8 pieces of genetic material that code for somewhere between 10 and 12 proteins. There's a little bit of discussion about that, but we wanted to find the ones that were most likely to be the same across all influenza viruses, since we were not sure what kind of virus what subtype this one would be. So my
Click in the middle of your PowerPoint. Use your
Or you maybe have to go down to the bottom left. There may be an arrow there.
yeah, I'm having a hard time controlling my trackpad because I can't see. Let me stop sharing and share again, if I can even do that.
Sure.
My my cursor is not showing up, which makes it very hard to know where I am.
That would.
Gosh, how frustrating.
There you go.
I don't know if I can do that again though. So, all right. So back in 1995, there was not, at least we at the AFIP did not have software for aligning sequences by computers. So what I ended up doing was typing out the sequences, printing them in a proportionate font, and then lining them up by hand and identifying places where the sequences were different, with the idea of designing primers in places where they were the same. And then there was just that picture there of me with Jeff Taubenberger and holding a autoradiogram. So that was how we did the sequencing. All of that is unbelievably antiquated now. In fact, Jeff can now sequence an entire influenza virus in one deep sequencing run. And it took us 7 years to sequence the virus, and you can literally do it in a day now. So there were all of those practical questions that we had to deal with. But there are also some existential questions. Where did this virus come from? Being the key one. Because of course, if we knew where it came from, maybe we would be able to prevent another flu from emerging the same way. And then why was it so lethal? It was dramatically more lethal than even contemporary viruses. Influenza used to kill a lot more people than it does now. Again, because people who caught influenza were so susceptible to getting a bacterial pneumonia afterwards, and there were no antibiotics yet. But even so, the 1918 flu was really probably about 7 to 10 times more lethal than a contemporary, 1915 to 1917 flu. The other thing that was really unusual about the 1918 flu, well, actually two unusual things. The graph on the top left shows the course of influenza deaths from June of 1918 through June of 1919. And you can see that there were 3 distinct waves of influenza. And that's quite unusual to have 3 waves in such a short time. There is still some controversy over whether that first wave or all of that first wave was actually the same virus. Nevertheless, it was a lot of flu in a 1-year period. The other thing that was very odd and has never been seen again, thank goodness, about 1918 was the age group that was most susceptible to severe complications. It was quite a sharp age incidence difference for people who were between the ages of about 25 and about 35. The dashed line on that graph is the average age-specific mortality for 1911 to 1917. You can see it's high in babies under 1-year-olds, and then it starts to rise again at the above 64-year age group and gets higher and higher. That's the same pattern we see now with coronavirus, that the death rate is higher the older you get. But 1918 had this really odd hump in the middle, and people talk about this as a W-shaped curve instead of the typical U-shaped curve. One thing that is interesting about it that we spent a lot of time puzzling over was that the death rate for everybody up to age about 70 was higher in 1918 than the average flu before that. But for people older than about 75, the death rate was actually lower in 1918 than in the average year, which we still assume, probably meant that people over that age group had had some exposure to the same subtype of virus, and so they had some limited amount of immunity to it. I mean, obviously the death rate was still very high, but not as high as from a regular seasonal flu. So in our search for more information about the 1918 flu, we came across another book, and that book was by Dr. E.O. Jordan, who was a very prominent microbiologist. He was actually one of the founders of the American Society for Microbiology. This little plaque is in the lobby of the American Society for Microbiology. He was asked by the American Medical Association in 1923 to write a comprehensive literature review of all of the research that had been done on the 1918 flu. This book was the result. It was published in 1926, I believe, called Epidemic Influenza. And this, along with Crosby's book, was our Bible of finding out what was going on in 1918. Jordan was clearly brilliant. I don't think we ever asked a question about 1918 that we didn't find he had thought of in this book. He wasn't always able to answer it. They were not even able to identify the causative agent of the 1918 flu in 1918 because they were not yet able to isolate and cultivate viruses. I took a little picture of the last 2 chapters from the table of contents because they're so salient now. And he asked all of these questions. The preventive measures, he delved deeply into communities' experience with what we now call social distancing and quarantining, whether closing schools made a difference, whether prohibiting public gatherings made a difference, whether face masks made a difference, whether having had the virus in a previous wave gave you immunity, whether you could develop a vaccine. And the vaccine section is haunting because they didn't know it was a virus. So they were vaccinating people with various bacteria that had been isolated from people's lungs, and clearly none of it worked. But what was stunning about that section, and really the whole book, is Jordan's unbelievable ability to avoid confirmation bias, that any result that he would report, he would also report what the weaknesses of that research were and what other things might be considered. He's just really such an impressive scientist. And I think we have a tendency to look back on scientists of the past and assume we have nothing to learn from them because we know so much more now. But when it comes to modeling really sound scientific judgment, we all have a lot to learn from people like Jordan. You can see that he also looked at whether there was any clear relationship between the weather and the spread of the flu. You know, we've heard that with coronavirus. Will the summer months cause it to go away? Humidity, seasonal variation. It's really stunning, the number of questions that Jordan asked and tried to answer. So some of the things that he was asking were when and where were the first cases of this influenza. There were certainly outbreaks in the spring that had characteristics that make us think they were probably the same virus. They had the same high mortality rate in an age group that normally doesn't die. But that was an open question at the time and remains an open question, is whether the spring wave was caused by the same virus. Was that unusual age-specific mortality the same everywhere? Because there was a lot of speculation that the reason so many young people died was because we were in the middle of World War I, and maybe this is just a problem of soldiers all being crowded together, and that's why there were so many deaths in that age group. But that turns out not to be the case. That age-specific mortality was extremely consistent across the entire world in all communities, rural, urban, everywhere, and in all ethnic groups. So they wanted to know, why did so many 25 to 35-year-olds die? And the very simple question of how many people died. There's a whole chapter in Jordan's book where he goes through all of the mortality statistics from every country in the world and added it all up. He came up with 20 million, which was the number that people used about the 1918 flu for a long time. Subsequent research suggested that that was probably an undercount, and the number was revised upward to about 40 million. I think that's the most solid estimate, but you see people, you see estimates up to 100 million, even more than that, that are based on no evidence as far as I can tell, other than they must not have counted right. But I think 40 million is probably a really sound number, and that's bad enough. It doesn't need to be 100 million to care about it. So just another couple of examples of the thoroughness of Jordan's work. The map of the United States is trying to track the spread of the virus. So the different hash marks and dots and shadings are all correlated to dates when the virus reached epidemic proportions in different parts of the country. On the right, this is again, just looking at, have we ever seen this age-related mortality bump in the 25 to 35-year-olds ever before? And there's all of the years leading up to 1918, carefully measuring, those numbers and showing, nope, it's a W. It's always a U. It's always a U, until 1918 when it was a W. So the answers to those questions that Jordan and his contemporaries were asking were important, of course, and they were still important in 1995. We were trying to figure out, could something like this happen again? Where should we be monitoring? What should we be monitoring for? Can we prevent another pandemic from emerging? And how best can we prepare for the emergence of a new influenza virus? What we found, because we were eventually successful in sequencing the entire virus, was that the 1918 virus seemed to have jumped directly from birds to humans without any reassortment. So let me just unpack a few of those words and a few of those concepts. Influenza virus is endemic in wild birds, mostly ducks and geese. It's been found also in a few other birds, seagulls, but ducks and geese are really its natural reservoir. There are dozens of different strains in birds, and they don't cause the birds to become ill, the wild birds. They just carry this and pass it from each other, one to the other, through the water. It's an intestinal virus in wild birds. Again, there are 8 segments in the virus, and if a cell is infected by 2 different strains of influenza virus, those 8 genes can kind of mix and match and come up with new viruses that have some genes from one strain and some genes from another. This didn't look like that had happened. At least if it was a reassortment, it was between 2 avian viruses. There was none of the segments seem to have any of the changes that are characteristic of human viruses. There were also no changes at the antigenic sites on the surface of the virus that change most quickly when the virus is circulating in humans in response to evading human antibodies. So it didn't look like it had been in humans for very long. Where a big theory at the time was that the flu had gone from birds into pigs and then from pigs to humans. We can't prove definitively which direction it went, but if you look at most of the evidence of outbreaks, it seems that it went from humans into pigs and not vice versa. We also found that there was probably a major drift event in that virus by about 1921, which is not surprising because by then every single human being on Earth had been exposed to this flu. And so in order for the virus to survive, a variant that did not, that evaded all those antibodies to the 1918 strain, there was huge selective pressure for that, and it looks like it happened. The best evidence of that is there was a major serology study in the 1930s, and it's amazing how many things are similar to what we're trying to do with the coronavirus. What they found is that anybody who had been alive in before 1920 had one kind of H1N1 antibody, and anybody who was born after 1921 did not have that antibody. They had a different set of antibodies. So the virus changed pretty dramatically in 1921. And the other thing that we, I guess, maybe foolishly assumed is that if we sequence this whole virus, there would be something really obvious about it that would explain why it was so lethal. And that was not the case. There was nothing about the sequence that seemed really unusual. So in 1995, what were people worried about? There are periodic pandemics of influenza. I've got the years listed there. You can see they're usually about 20 to 30 years apart, as little as 11, as much as 39. But by 95, people were really starting to feel like it's time for another influenza pandemic. And so people were nervous about that. Adding to that nervousness was that in 1997, there was an outbreak of avian flu in the bird markets, the markets of Hong Kong. So this was among chickens. It's incredibly pathogenic in chickens. It just wipes out chicken flocks. And so the abbreviation is HPAI, highly pathogenic avian influenza. This was a virus that went from wild ducks or geese into chickens. And since the 1918 strain looked avian, this seemed like this may be a place where the next influenza pandemic will come from wild geese into domestic poultry. We were learning at that time a lot more about the natural reservoirs, the kinds of strains that were being carried in ducks and geese, and how prevalent it was. So that was sort of, you can see the line of thinking there was, this is where we should be looking for the next influenza viruses, in these bird markets. Now, since 1995, we had that one book by Alfred Crosby. Since 1995, lots and lots and lots of writers have written about the 1918 flu. As I said, I still think Crosby's book is the best one, but many of these are excellent as well. Gina Colada's book in particular does a really good job of looking at the search for the 1918 flu. She covers our work, but she also has an absolutely wonderful chapter about Johan Holten, who I'll mention in a couple of minutes. We also had an outbreak. It sometimes is called another pandemic. In the 2009 H1N1 strain, it was a pandemic in the sense that it was a new surface hemagglutinin, and so it caused a very large number of cases. It was extremely mild though, and didn't cause very many deaths, which is another thing that people often sort of equate the word pandemic with high mortality. In this case, it was not high mortality. This slide is too busy for you to see all of the details, but the important thing about it is way over there on the left of that virus that's all gray. It jumped, you can see that first little short arrow. We assume an avian virus jumped into the human population. That's the 1918 flu, that's all red. That went from humans, as I said, into swine, the virus above it, that's also all red, but kind of a heavier red bars. And then if you keep going down and you get to the next yellow arrow that's a little bit longer, that's the 1957 pandemic. And you can see that the green squiggles are new genes that came from an avian source into the human population. But you can see that that virus retained several genes from the 1918 virus. So it had 5 1918 genes and 3 new ones. And then in 68, you can see on the bottom line there, you still have several genes from the 1918 flu. You still have 1 gene from the 1957 flu, and then you've got 2 new ones. This is all just to say that the influenza virus has a lot of tricks up its sleeve for generating variants that can continue to spread in humans. And then this 2009 reassortance right there in the middle, sort of to the right of the middle, ended up having gene segments, still got some from 1918, still has 1 from 1957, still has 1 from 1968, and then it got a couple from and the swine flu. So the influenza virus really, really can change a lot. So what was our policy response to all this that we were learning about the 1918 flu and subsequent influenzas? Well, we started doing a lot more monitoring of poultry markets. An international early warning network started up in the 90s called ProMed. It's still around. It was just a very simple listserv that gathered information from doctors all over the world who could write in and say, hey, I'm seeing this weird respiratory illness that I haven't seen before. I'm seeing this weird sexually transmitted disease that I haven't seen before. And ProMed monitors those, verifies those, makes sure there's more than one source of information, then issues an alert. It was successful in detecting SARS. The first SARS cases were reported on ProMed. The first MERS cases were reported on ProMed. Ebola. It's really a fantastic sort of cloud-based or crowdsourced surveillance system. We have put a lot of effort into figuring out how to detect and respond quickly to poultry outbreaks. We've done more research into natural reservoirs. And there's been a lot of research into universal influenza vaccines because this virus can change so rapidly and emerge so rapidly that it seemed like the best public health response would be to have a vaccine that would protect us no matter what influenza strain emerged. So all of those policy responses, I think we can say that they've had a lot of success. These highly pathogenic avian influenzas have never managed to jump into humans. They have, at least they haven't jumped into humans and then been able to spread from human to human. There have been human cases of HPAI, but it hasn't then spread within the human population. And as I said, ProMed has been enormously successful, and the universal influenza vaccine work is showing some promise, although it's not yet produced a proven vaccine. But what did we miss? Because we were so focused on the idea that the big problem, the big pandemic was going to come, it was going to be flu, and it was going to come through somehow through birds. We weren't really monitoring these wet markets, these live poultry markets for non-influenza viruses, or for that matter, non-poultry markets. this ProMed system is incredibly successful at detecting outbreaks. But the next step of saying, all right, all of us public health agencies around the world need to get together and figure out what are we going to do when ProMed detects something. Every time it detected something, it was sort of an ad hoc reaction. In retrospect, we should have been saying, we need to be ready for whatever ProMed throws at us. And I think also there was maybe some complacency because when ProMed identified SARS and MERS, they were contained relatively quickly. So I think that there was some thought that what we have in place is working. Clearly, what we had in place was not quite enough. And so that just led me back to this sort of philosophical idea of how do we decide what we know is the most useful? And how do we decide what we don't know, what's the most important thing for us to figure out? And obviously that's a very tall order to say, what is the thing I'm not thinking of? That's not, there's no guarantee that you're then going to think of the right thing. But I do think going back to reflect on, am I so sure that the path I'm on is the right one that I don't need to think about other things that might be coming at us. And then I just wanted to do this little bit of a postscript that our first paper was published in Science. It was based on just a few hundred base pairs of information from the 1918 flu that we had managed to extract and sequence from one of those pathology samples from the AFIP. We had only found one positive sample. We only had a very small amount of material from that. So it was not at all clear we were going to be able to sequence the whole virus from that sample. But in '97, we were contacted by Dr. Johan Hulten, who was a retired pathologist, and he had tried to get his PhD in virology in 1953 by going to Alaska and exhuming the bodies of Inuit victims of the 1918 flu and trying to culture the virus from those samples. He was not able to do so. The virus is very highly deteriorated even in those samples. He ended up dropping out of the PhD program as a result and becoming a pathologist. But when he read our paper, he offered to go back to the village and get permission to exhume the bodies again and send us samples, which he did. And 7 years later, Johan and I traveled together to Brevig Mission, which is the name of that little village, to thank the community for their help and to commemorate the dead. We put up a plaque with all of the names that was affixed to the cross that marks the mass grave. And we thanked the village. This is Johan there on the left with the white beard. He was in his 70s at the time. He is still around. He lives here in San Francisco. He's in his 90s now. Quite a remarkable man. And then one final story. The crosses there are the crosses that mark that grave in Brevig Mission. You can see the plaque that we affixed to the cross there. At some point, I don't remember when we found this story, we found a book of stories from Alaska, and we found this one called Alone with Death on the Tundra. It was written by a woman named Clara Faso, who was married to the pastor of the village of Brevig Mission. He was a missionary to the Inuit. And she tells the story in this essay of being the only adult in the village who did not come down with the flu. And the flu was brought in by a mail carrier on a dog sled. He fell ill soon after arriving, and within a week, every single adult in the village was sick, and 65 out of 80 adults in the village died of that flu. So to find this story that was about the exact village where our case had come from, that some of the people she wrote about, one of them might have been the body that we got the sample from, it was really haunting. Amazing story. So it was really a genuine privilege to work on those samples. It was illuminating and inspiring to read the work of the scientists who were working, scientists and physicians who were working so hard in 1918 to stop the epidemic and then to figure out what had happened. And of course, to just remember the millions and millions of people who died in 1918. So I'm just a quick thank you to Jeff and our AFIP colleagues who worked for 7 years to sequence the virus. Of course, to my colleagues at NCSE who indulged me in a great deal of talk about the 1918 flu, although now they're more interested in it than they used to be. And the whole public health research community who have worked so hard for all these years to try to be ready for what we're going through now. And of course, all of that research has to be paid for. And I also just want to give a shout out to the journalists who let the world know about this and the historians who've studied these flus and told these stories and gave us the gift of the evidence that we needed to do our work. So I will stop sharing if I can. I still can't see my cursor.
People would like you to put back the books, if you could find the slide and put that up with the books.
That one?
That's the one. Thank you.
Okay.
Let's see. All right, Joe, you can unmute now if you wanted to say something.
I want to say thanks. That was a great talk. Really, really enjoyed it. And you should join us more often. Thank you.
It's very kind of you.
And I want to say thank you, and I'm sorry for not being able to continue hosting. My internet has gone to almost nothing. So I'm going to hand this back to Patricia.
It's some evening here. My nose luckily has stopped bleeding, so you can hand it off to me, and one way or another we'll get this done. I think there are a lot of comments and questions, so people can feel free to unmute themselves if they want to ask. It looks like, Neil, did you have something to say?
Yeah, I just asked if there was any new insight that Ann's aware of into the genetic basis of the increased virulence of the 1918 strain.
So I have been out of the lab for 15 years, 16 years, and I haven't followed all of the work really closely. My understanding, though, is that there seems to be a mutation in the PB2 gene that seems to be correlated with virulence. But I'm sorry, I can't give you any more details than that. There's a lot of work going on in that.
Yeah, it's been my impression that it's been extremely difficult to try to pin it down on any one genetic change or even any one gene.
Yeah, yeah.
I think that's informative because it tells you that virulence is a very complex business.
Exactly. And the other thing that I think it tells us is that, so there was a lot of, Jeff and I spent a lot of mornings drinking coffee and talking about that age-related mortality curve that's so
Right.
weird, and trying to just figure out whether that had something to do with the virus or whether that had something to do with the age of the people and their previous exposure to influenza.
Right.
And what you're saying about trying to pin the virulence to a genetic marker might not even be where the answer is. The answer might be in their immune response.
Well, it's probably some interaction, but
Yeah.
But my point also is, I mean, this is a virus with, like you said, 10 to 12 genes. Now imagine a bacterium with several thousand and trying to figure out the virulence of a bacterial strain.
Yeah. Yeah. Thank goodness the 1918 flu is only 15,000 base pairs, or I'd still be sequencing it.
Jürgen, I think you had a comment earlier or a question.
Yeah, so I was wondering about the W shape, if the bump or the hump in the middle, is that due to soldiers being basically not in a good condition when they came back or going to World War I and being more susceptible to influenza?
So that's a great question, and as with all the questions, Dr. Jordan looked into that and came to the conclusion that although the flu spread extremely well in barracks, that was a perfect environment for spreading the flu, that age-related mortality was consistent in every community. So we actually looked at the death records for the state of Kentucky and compared the mortality, age-related mortality rates in the camps. There were 2 big Army camps in Kentucky, and it was exactly the same. You know, men and women, soldiers, not soldiers, that age group just was very susceptible.
One thought had been that there had been a flu that had run through 30 years or 20 years earlier that gave some immunity to the older folks. Does that have any merit to it?
Well, I think the evidence, it all has to be indirect evidence because we are
Sure.
never going to actually get those samples. It does look like people born before, let me see if I can remember my numbers right, born before 1860 or so, that probably an H1N1 strain had been circulating before 1860. And then there's pretty good evidence of a pandemic in 1890. And so that would have been 28 years before 1918. So that correlates kind of, sort of, to the higher death rates in people in 25 to 35 years old. None of the numbers add up perfectly. So it's hard to come up with a really clear, a really great hypothesis that sort of fits all of the data that's out there. There's also been a lot of talk, and we're hearing this term thrown around now again, called antibody-dependent enhancement, which is known to occur with dengue virus, that there are 3 strains of dengue virus, and if you get them in one particular order, when you're infected the second time, you actually get much sicker. The antibodies to the first strain actually make you more vulnerable to the second strain. And that's never been reported in influenza, but that would really, that would be a great explanation for that enhanced death rate in that age group if they had had a strain that made them more vulnerable. But again, it's never been seen in influenza, but people are a little worried about that with this coronavirus, that when we say that people who have, if they have antibodies, then they're immune. Yeah, maybe, but they also might be more vulnerable. So there's still just so many open questions about that.
Dorothy Eller has a question. She says, is it the same flu that was called the Spanish flu? And if so, she thought that the Spanish flu came in 2 years. There was 1917 and 1918 was the second wave.
The 1918 flu and the Spanish flu are the same thing, but it was in 1918 and 1919. So the first reports were in the summer of 1918, and the first kind of major wave in Europe was in the summer of 1918, and the Spanish was the only, Spain didn't have censorship of its press because it wasn't fighting in the war, and so that it reported on the virus, whereas France and Germany and England and Italy that were in the middle of fighting the war, they didn't report on it. So it started to be called the Spanish flu because people thought that's the only place it was, but that was not correct.
All right. There was Kingman. What did you want to say?
So you mentioned the secondary pneumonia as a cause of death. Was that common? I mean, here in this virus, you get really a tropism to the lung with ARDS relatively quickly, but a secondary pneumonia would take some period of time.
Yeah, and the most common course of the disease in 1918 was to get the viral pneumonia, start to recover, and then, in a week to 10 days, coming down with a much more severe bacterial pneumonia.
Right.
According to the John Barry book, anyway, most of the scientists at the time were convinced that it was a bacterial infection, and were going after the bacterial influenza.
Yeah. Yeah. Well, they found bacteria in almost all of the lungs.
Although it was in every case, so they thought, yeah.
But even then, they realized that it wasn't the same bacteria in every case, and that it tended to be, for example, on Army bases, most of the soldiers in one base would die of one bacterial infection, but on other bases, a different one would be predominant. So they knew at the time that it was probably not bacterial, but again, they had no ability. I mean, the idea that there even were viruses was still really new, and the ability to actually work with them was zero.
By the way, the tropism for flu is based on recognition of sialic acids on glycoproteins on the cell surface. So I don't know that there's one specific glycoprotein that serves as a receptor. So it's different than a virus like the new coronavirus, which has a particular protein target.
Mm-hmm.
Vivian, you had a question?
Government in handling the epidemic, the response.
Could you repeat it? It didn't come through very clearly.
How active was the government in handling the response to the pandemic?
Well, you know, the federal government had much less ability to act at scale and at speed. You had telegraph then. That was the fastest word could travel. So it generally it was up to city and state governments to respond.
They didn't even have flip telegraph. But the government, since we were at war, they downplayed the whole effect, and really probably contributed to the effect. We had some reports from the Cleveland Public Health Service that said that they essentially closed off all weddings and funerals and closed down the restaurants and bars, just like we're doing now.
Yeah, and it was, that response was varied across the country, and I think a lot of people are now digging into, really trying to see whether those measures made a difference. Jordan, of course, looked into that very carefully, and this is where, again, I find so impressive his ability to avoid confirmation bias, that he would constantly say, okay, so somebody said that Philadelphia had this terrible outbreak because they had a parade, and San Francisco didn't have the first outbreak because they had everybody wear masks. And he would, point out that there were communities that were otherwise completely identical. Like there were camps of workers of various kinds, miners and people doing major construction projects were in the same town with exactly the same kind of quarantining approaches, where one group would have a death rate 5 or 6 or 7 times greater than the other one, when it looked like they had done exactly the same thing. So he just, a lot of caution about choosing one metric and saying, this is probably the thing that made the difference, because you can always find counterexamples.
Let me just change a little bit here. Gordon Maupin, did you have a comment to make? Do you want to ask your question? Should I unmute you? Yeah, you got it.
Yeah, this is more for your current job, Ann. NCSE fights anti-science on evolution and climate. I was wondering if NCSE sees an opportunity with COVID-19 to fight anti-science on a broader front. I was particularly taken by an article I saw today about the anti-vaxxers already gearing up to fight should a vaccine become available.
Yeah, We have generally stuck to areas of science that are misrepresented in the classroom. And since, vaccination is not part of the normal curriculum, we don't have a situation where legislators are saying kids should learn that vaccines are bad, and teachers who are teaching kids that vaccines are bad. And so we haven't focused on that. We have been writing what we're calling a teachable moment every week. A lot of the science teachers who work with us have been asking us to help provide information and activities for their students about the coronavirus. So that is published every Friday. You can sign up for that newsletter at our website. And I do think that we may have to get a little bit involved in the vaccine battle if it does start to take off. But our constituency as science teachers, we have not tried to influence overall public opinion. That's a pretty heavy lift for an organization that only has 10 full-time employees.
Let me give you a quick shout out. I'm a member of NCSE and think everybody listening should support NCSE.
NCSE.ngo, man. Go and join.
Definitely. All right. Well, I wish we could let this go on further, but it is getting a bit late. So if we could all thank you again so much for this. It was fascinating.
Thank you.
Thank you.
Great questions. Wish I could see you all.